Good afternoon, everyone. Welcome to the third session for the day. It's an honor to welcome Professor Vaibhav Tripathi from Cognitive and Brain Science Department of IIT Gandhinagar. Starting in computer science engineering, Professor Tripathi transitioned to neuroscience through a master's in computational biology and a PhD at Boston University, exploring attention and memory. He continued as a postdoc at Harvard, studying large-scale brain networks. Bridging engineering and neuroscience, his work brings a unique perspective to understanding the brain. Please join me in welcoming Professor Vaibhav Tripathi.
Hello. Audible at the back? No? Okay. Audible better? Low? अच्छा ठीक है अच्छा okay audible now bass voice okay how many of you are engineering students here? How many of you not engineering students? Brave ones, nice. I hope you're having a good time, able to understand it well. Okay, nice. How many of you are bachelor students? Masters? PhD? Okay. So no, bachelors had so much energy. Masters, PhD like thak gaye hai sir, subah se kaam karke. Okay, you had heavy lunch, I guess. Lots of chawal and all. No? So I'm pretty sure that you guys will feel sleepy sometime. If you do, I'll not blame you. ठीक है. ज़्यादा नींद आ रही है तो सो भी जाना यार. But okay, so today's topic is connectivity-based predictive modeling. But this is the small title. This is the whole big title: MRI functional connectivity-based predictive modeling of brain organization and cognitive state for healthy and clinical population. So already my introduction is done, so we'll start here, which I don't think. So that is not updating. Yeah? Okay, it's taking a bit time. Okay, so why to study the brain? We'll go over that, how to study the brain. Then we'll talk a bit about brain connectivity and activity. We'll talk about two different techniques, predictive modeling techniques to model the brain. I assume a lot of you have not studied brain before. How many of you are aware with neuroscience in general? Okay, few hands. How many of you are not aware of it at all? Okay, तो इतनी ही श्रद्धा से जैसे हाथ उठाया है क्वेश्चंस पूछना. There is no tax for questions. ठीक है, समझ में आया? Yes. And there is no stupid question. Stupid questions are जो आपके मन में चल रहा है और सबके मन में चल रहा है. आपको लगता है अरे मैं कैसे नहीं पूछूं? No, feel free to ask. ठीक है. I'll not cover as much material, but we can ask more questions. Idea is that you learn something so that आप आगे जाएं और आपको अच्छे समझ में. अच्छा, English comfortable है सब, हिंदी से comfortable है सब. People are not comfortable in Hindi at all. ठीक है, 1, 2. So I'll try to be speaking in both. अगर if I'm speaking in Hindi, I'll also repeat in English. ठीक है. Sometimes joke are better in Hindi, you know. So just can't it. Okay. So why to study the brain? So you're doing a thing on AI, and so brain-inspired models like convolutional neural networks, LLMs, were based on brain organization. So you see this network here, this one. Okay, this network here, I cannot see, maybe. Can you see my pointer? Yes. So if you see this network, this was something that was, after years of investigation, these two guys consolidated this all into this very deep kind of architecture in 1991. And this is how the visual system was organized. So when the first convolutional neural network was born, it was based on this visual system. Okay, we stack layers just like they are in the visual system. And in 2012, something amazing happened. Can you guess what? You have to speak louder. Hey, go ahead. अरे आपको ही कह रहे हैं. ImageNet, yes. ImageNet happened. Convolutional neural network trained through GPU became a reality, and the boom just happened. But the ideas, the principles in neuroscience were there. The other principles in neuroscience that are very highly used in machine learning, can any one of you guess what are the other ideas from neuroscience which are used? Language processing. So large language models, the idea behind LLMs is next word prediction is in the similar way the brain also works. Any other models? Neural nets तो हो ही गया ना, convolutional neural nets में NNs क्या है? Okay, or anything else? So reinforcement learning is also derived from the dopaminergic system in the brain. So reinforcement learning works through that. So brain inspires AI, but AI can also inspire brain too. That's why AI and neuroscience need to work together in conjunction. AI can inspire, deepen neuroscience, and neuroscience can. So I guess you might have seen this video. How many of you know what this is? So this was a recent thing from Neuralink. Neuralink, how many of you have heard about? Yay, enthusiastic hands. So Neuralink is a company by the super villain of our times, Elon Musk. I will not say that, but they are creating this amazing tool which is collecting data directly from the brain. It's not something that is new. It has been done for the last 40 years, 50 years. They are doing it at a scale, and they are trying to make it very robust. So now they have 10 patients. These paraplegic patients who cannot move their hands and legs, they're putting electrodes in different parts of the brain, getting their recordings, and allowing them to talk, move, operate a cursor, play games and all. So this was a, let me see, how can I do this? So this is a monkey playing a Pong game, and it's controlling one of the Pongs just through its brain activity. And it's getting a juice. So you see, it's holding something, so it's getting a juice reward. So this monkey was not fed for some time, it's very hungry. So all it has to do is move this thing so that it can operate. Now on the left, what you see is brain neural activity on your, yes. Or it's using to decode where to move. And this is from the motor cortex of the monkey, which is responsible for motor commands. The monkey is just thinking about movement, not moving any of the hands. It's just focusing on the juice now. Okay. So this is where they would like to go. Again, this is from the Neuralink studies, but they want to do it for humans. And I think they are on a good track. They need a lot of work, but they're doing pretty good job. So where can neuroscience go with all this? Once we understand, one, how the brain works, it can help us define these tools, create these tools, which can help in the clinic for rehabilitation, for different disorders, for different diseases. So very understand to think about the brain and technology in conjunction. We understand how the brain works, understand how the technology works, and it's a loop that goes around. So now I've kind of established the applications. I want to like to go behind and see, like, ask the question, what is neuroscience? How many of you know what is, that's already there. The neuroscience is the set nervous system, brain, spinal cords, peripheral nerves. So the central nervous system is composed of the brain and the spinal cord, and the peripheral nervous system on the sides is composed of these nerves, ganglion, etc. Neuroscience is a highly interdisciplinary field: biology, psychology, computer science, philosophy. I myself was a computer science major, and like, I want to study the brain. I was in ACM for a while. I was like, bahut ho gaya. I want to understand the brain now. So been 10 years in neuroscience, and it is fun. And I'm still, my work is very computational, but also appreciating the biology. And biology is beautiful, you know. A lot of times in our 11th and 12th, we have to make this decision: maths le ki bio le. Bio loge to doctor banoge, maths loge to engineer banoge. But the samagam, the bringing them together, is amazing. Mathematical biology is beautiful. And like, once you understand how the body is functioning, you can use those principles to understand better, create better tools and better systems. So there are many subfields of neuroscience. We start from different levels. So going from molecules to the whole organism. So molecules, you do what is called the molecular neuroscience. You understand different chemicals, how they work. Then there is a cellular neuroscience at the levels of neurons and synapses. Then there is a systems neuroscience where you see in system level understanding how different brain regions are working together. Cognitive is on the whole, like how the brain is working at a big level. Computational, we use computational modeling, try to understand how these neurons are working. The idea, what's the basic idea in computer science? What is a computational unit in a circuit? The lowest computational unit in a circuit, what is that? If you take a circuit, microprocessor, what is the lowest computational unit? Gates, very nice. And which is the most popular gate we use? NAND gate or OR gate. These are universal gates. We can design the entire system. But that is not how the brain works. Brain works in very different. We'll talk about it more. So computational neuroscience is trying to understand how the brain works. Can we find principles of the lowest units and kind of create from the bottom up? Clinical is application to clinical aspects, and neuroengineering is again the BCI that we saw, the monkey playing, is something we call as neuroengineering. So we'll go a bit about brain anatomy because today what we'll be discussing is the application aspect, but thoda sa baseline bhi chahiye. So you guys might already know about this. The brain has major components: cortex, cerebellum, brainstem, spinal cord. There's a central system, peripheral system. There are different lobes of the brain: occipital, piche ki side, parietal on the top, frontal in the front, on the side. There's something called as cerebellum. There's our different regions, and different tissue types, different regions of the brain are associated in different kind of functions. Okay. Again, lobes of the, we said, different kind of functions. So the visual region is the visual hierarchy you see from here to here. This is called the dorsal visual stream. So in we have these regions from V1, V2, V3, V4 till MT, and this is what convolutional neural networks used. But there's a lot of tissue types here which have their own way of optimizing the world. There is a specialized face region in the bottom of the brain, and there's like different aspects of the brain which are very, very interesting. But how we can use that, it's not well known. People are still working. There's an entire field called NeuroAI. Our lab, including labs around the world, are using principles from the brain to understand how we can optimize or do neuroscience better. So going into the raw, this thing of how the brain works. So these are some core entities in the brain. So neurons and synapses. So neurons communicate via electrical impulses. So neurons are the core structure. They are the core cells of the brain. So neurons has this body, there's a cell body, there's an axon, and there's a axon terminal. And at that terminal are these powerful things called the synapses. If you zoom in at this end, there's a powerful thing called the synapses where information is transformed from one neuron to the other. So it's very beautiful the way this, so this is all neurotransmitters which kind of jump from one to the other, and the entire way in this which signal goes through action potential is very beautiful. It's one of the most beautiful parts of biology. There are very interesting videos online. Wo chahe to search kar lena. It's something happens. Let's say maybe it's a mouse or maybe a spider. The brain detects और वो क्या करता है कि some sodium ions from the outside of the cell jump into the cell, and that triggers this entire thing which is called a spike. So spike is the most basic unit of brain. Whenever a spike happens, there is some signal that is happening. Now brain is composed of millions of spikes going around like firing, and trying to make sense of that neural code. There's a lot of work that people have done to really make sense of that neural code. In addition to these action potentials, which are one-to-one signals, there are things called neurotransmitters. So we have different kinds of neurotransmitters like adrenaline, noradrenaline, dopamine, serotonin. So can you name an activity where dopamine is released? Exercise. Can you see? Achha. What else? Where dopamine is released? Social media. How many of you watch reels nowadays? Reels, YouTube shorts, short form content. कितने लोग नहीं देखते हैं? अरे वाह, क्या बात है, आप लोगों को अवार्ड मिलना चाहिए. बाकी, so when you see reels, what happens? Scroll, और आधा घंटा कहाँ चल गया पता नहीं चला. Your dopaminergic system is hijacked at that time. Your brain is giving you these high reward signals. और देखो. और जैसे एक ड्रग एडिक्ट होता है आपकी वैसी हालत हो जाती है. लाइक आपको you can keep on getting shots of this thing. So dopaminergic system is that neurotransmitter. How do neurotransmitters work? They are broadcast signals. Action potential 1-to-1 connection. 1 to many, maybe 1,000 neurons or so. But अगर आपको broadly broadcast करना है, you give these neurotransmitters. एकदम जैसे आकाशवाणी में वो होता है, AIR पे बोलते हैं. Something like that happens when you use these neurotransmitters. So it's very amazing, like understanding. So each we talked about this here or here. So each connection, each synapse, what we say, has a certain specific neurotransmitter. Or ye neurotransmitter when they release. So when people take, so there's a very interesting video. How many of you know this YouTube channel called something like that, in a nutshell? So there's a video in it how heroin works or how fentanyl works, something like that. आप वो देखना, the amazing video of really how the dopamine system just hijacks your entire brain. These neurotransmitters are very powerful form of communication. They are a way of the brain to like force the organism to do something at one time. Why it is needed? So when is adrenaline released? Can any of you tell? Fight or flight, or अच्छा, that are adrenal glands. Adrenal glands produce adrenaline, but adrenaline are released either in a fight or flight situation. आपके सामने एक कुत्ता आ जाता है, भागने लगता है, then either you shoo it away or you run. Hopefully you don't run. यहाँ के कुत्ते थोड़े पागल ने, देख लेना भैया, भागना मत, ठीक है. तो what will happen is this adrenaline releases like a flurry in your brain and signals the entire thing that I should run now or I should fight. ठीक है. So these are broadcast signals. Now the brain is very beautiful. So this was a recent app that came. This is, can you guess what this is? Any guesses? Brain of a? Okay, very close. Can you guess what insect? Huh? Fly. Nice. So this is the brain of a fly. What fly? It's called a fruit fly. कभी आपने देखा है? You put bananas outside. You have these small flies going around. That fly, it has around 1 million neurons, maybe more, and they were able to track all the neurons. And it's such an intense map. Each neuron is connected with 10,000 other neurons. The computational ability in a fly's brain is much more than what we can think of it is in a XAI ka GPU farm of 100,000 GPUs and all. Okay. So the brain is amazing. If you can understand how the brain works, there is so much you can do. So now they have this entire detailed map of fly brain. So agar aap jana, you try to search, they are now using it to emulate how a fly works. They have a very high resolution map in which they can show how the fly is working. So they are now trying to do this for mouse and humans as well. But there is a challenge here. Can you guess what that challenge is? Why we cannot do it for a mouse or a human now? Too much competition. What else? Size. Size of what? Size of? The difference in size. So this is that small and human are this big. So the complexity is, so this data set is 1 petabyte or about 2 petabyte. How much is a petabyte? 1,000 terabytes. So this is about a 1 petabyte of data, just one tiny fly. just multiply it 100,000 times. That exabytes of data we need just to model one human brain. Okay. So it's a computation, it's a limitation of our current technologies, but it's fun. So all this brain glob is organized into these things called networks. So brain mein there are some very well defined networks. One is what we call as a somatosensory network. So everyone please raise your right hand. Okay. वो थोड़ा सोचना पड़ा मेरा right कौन सा है. So the moment you listen to this instruction, you raise your right hand. This entire process give a coordination to your motor cortex, giving you this signal, I should raise my right hand. Your somatosensory and your motor cortex worked in conjunction, I want to raise this right hand. The moment you listen to this instruction, your cognitive control network got activated. कि क्या करना है, मुझे control करना है अपना हाथ. It gave a signal to the motor cortex that I should raise it up. How do you know that it's raised up? Your somatosensory cortex is sending a signal back, a feedback signal कि it is up. So you have all these sensors in your different parts of the hand, which will give you a very good कि मेरा हाथ कहाँ है, मेरी body कहाँ है in respect to like my particular this thing. There's this network in your brain called the default mode network. It's a very wonderful network. When was the last? How many of you are from Gandhinagar? Delhi. Okay. And what other places? Huh? You have to speak up. Okay. Where else? Acha. So when was the last time you went to, let's say people in Delhi? When was the last time you went to Delhi, India Gate? Think of the location you went to India Gate that last time, or any other place you love in your city. Just think of the time. So this particular process that you're thinking, mental imagery, what we also call episodic projection, thinking either in the past or the future, it uses this brain network called the default mode network. So default mode क्यों बोलते हैं? क्योंकि if you don't do anything, your brain kind of goes into this default mode. And sometimes it happens that when you eat food, it gets more activated. So maybe some of you are already started sleeping, but so I will not blame you. Your default mode is active. So the brain is a very dynamic network system, engages these different parts of the networks to do different things, and it's beautiful. So this is a map from my lab at Harvard, where we see that these different brain networks are not organized in a very clean manner. It's all a khichdi of different networks going in and out together. So we use a lot of different techniques to see where in the brain what is happening and try to make somewhat sense of it. So now talk about how we really study the brain. So we have a lot of experimental methods. So this figure kind of shows very nicely. So you have time on the x-axis and you have the spatial scale on the y-axis. So you can study at the level of the molecule at a millisecond level, or you can study at the level of the organism, social network level. RCB GT, people went to this crazy extent, हमको जाना है, so people study sociology, psychology, ये सब कैसे हुआ, that is at an environmental level. And then you're looking at the molecule millisecond level, fly के brain में क्या चल रहा है. So neuroscience covers the whole length and breadth of it. So my work has been mostly in around understanding EEG, fMRI, the organism level, so somewhere here, where the brain system and everything is working. Okay. But neuroscience is pretty deep. Here we have lots of way to like really look at the brain. But amongst these technologies, the one that are used in humans a lot, one is called invasive electrophysiology. We pluck open a brain, put some electrodes, see what is happening. The brain-computer interface that you see with the monkey, this is exactly what is done. The tissue is taken out, electrode is put in, and then we observe the activity of the neurons. कि कहाँ पे क्या चल रहा है. Let's say motor activity, तो कहाँ पे अगर motor region में चल रही है, visual region में क्या activity चल रही है, तो इससे we observe all that. There are other tools. One is called EEG on the left. So you put these electrodes on the top of your skull, and then वहाँ से we record various activities of the brain. We also have MEGs, which is magnetoencephalography. So your brain has this electric field lots of neurons are firing together. So what happens is it creates these very small electrical impulses, some millivolts, even microvolts, sorry, some microvolts, and it has an electrical signal and a magnetic signal as well. Ajay, why is that? Why does an electrical signal have a magnetic signal? What's the rule called? Okay. Who discovered this? Electromagnetism, किसको याद नहीं? Engineering people, no? Okay. कुछ याद आ रहा है, ठीक है चलो. So electric and magnetic, this thing couple, you don't right hand rule, current जाएगा, magnetic field होगी, so something like that happens. So your electrical signals go in this direction in the brain, so magnetic signal goes around this brain. So you can use MEG to kind of detect signals. And this is how signal looks like. This is time series data. On the x-axis, we have the time series. So we are recording from a signal, either EEG, MEG, or intracranial electrode, and that time series go up and down. Okay. So, and then we design a setup. Let's say I asked a person to move a hand, move your hand up or down, and then we see ki kya activity chal rahi hai. And that allows us to estimate ki, achha, which part of the brain is giving this activity, what kind of activity is happening, what brain networks or what brain rhythms are involved. So this gives us a kind of a detailed understanding of where it is happening, when it is happening, and what is happening. Okay. Then there's another technique called fMRI. How many of you have seen an MRI before or have had their MRI done? Okay. Oh, a lot of people. How is the experience? How was it the last time you went? It felt like it was nice. Okay, who agree with this assumption that this was nice? No, not a lot of people. So MRI is this big room. It's like nearly this size, the size of the stage. There's a machine with a small bore. You go inside, and the machine produces this beautiful signal, this beautiful noise, like tuck 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 tuck. The first time I went into an MRI 10 years ago, I thought the machine was malfunctioning. ये machine फटेगी. और मेरी पता है क्या चिंता थी कि देखो anyways I will die. The people who will clean up after this, वो इतना अजीब लगेगा ना, मतलब कैसे होगा? It was such a horrendous this thing, but now I can sleep like a baby. It's like the most soothing place. आराम से लेट जाओ, white noise आती रहती है, सो जाओ. It's hard for me to do a lot of these studies now. So my major study design is experimental technique is MRI, and what we get from an MRI are these sort of fancy blob-looking things. So various activities. Let's say I ask, you're listening to me now. So your language area in the brain starts to get activated. Some of sleeping with Big Basi, I can see. So some other brain region will be getting activated. Okay. When you're doing nothing, still your brain is pretty active. So it is what we call as resting state. Usme kya hai? There is some underlying activity happening in the brain. So it is not that when you're doing nothing, nothing is happening. When you're doing nothing, still the brain is very active. Still something very interesting is happening. Okay. So now here, what we want to do in this entire series of studies we want to explore, see how the brain connects, how different parts of the brain connect with each other, and how the activity of the brain, is there a link between that? So one of the big names in the field had this conjecture that each tissue of the brain has a unique connectivity pattern, and that connectivity pattern can help us predict function of the brain. So the idea is that if we can establish these patterns, define this connectivity, we can observe the activity. ये clear है क्या? Relationship between how the brain connects and what happens when it is active during a task or something. This is a very crucial point. इसके बाद अगर ये नहीं clear हो तो बहुत. Is this clear? How many of you it's not clear? Okay, ठीक है. At least he's on. So what is connectivity? Connectivity is different parts of the brain, how they are connected to each other. तो एक brain अकेले में काम नहीं कर रहा है. It is have these signals going to different parts of the brain, but there are some parts which are more strongly connected to one another than other parts. So once we kind of define how this connectivity is happening, we can then define how the function of different tissue types. जैसे मैंने अभी दिखाया पिछले उसमें कि इस part of the brain में we see activity of the language region. Maybe another the part in the occipital, what we call at the back of the brain, we see activity during visual task and all. So can we then link up these activities and relate it to how the brain connects? थोड़ा clear हुआ क्या? थोड़ा सा. Can you repeat? जो बोला मैंने repeat कर सकते हो. बोलो. उससे पूछा तुमसे, मैं उससे पूछा. हाँ. 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 हाँ, मतलब इसमें थोड़ा सा वो जब आप देखते हैं कि, तो हम ये देखना चाहते हैं कि ब्रेन कनेक्शन जो है ब्रेन का और ब्रेन की एक्टिविटी, उसमें रिलेशनशिप है क्या. तो द वे द ब्रेन कनेक्ट्स, एक्टिविटी प्रोपोगेट्स अलोंग दोस चैनल. बहुत सिंपल सा आइडिया. अगर एक रीजन में एक्टिव हुआ तो दूसरे रीजन को एक्टिव कर देगा. तो अगर आपने बस ये देख लिया कि ब्रेन कनेक्ट कैसे कर रहा है, तो आप ये जान जाओगे कि वो एक्टिवेट कैसे करेगा. दैट्स द ब्रॉड आइडिया. इज दैट क्लियर? नॉट क्लियर? You can ask. Loud. हाँ, तो very good question. तो कनेक्शन आप दे दो उसको भाई, अब बाकी लोग सुन लो.
I want to understand कि एक एक्टिविटी हुई,
हाँ.
तो कनेक्शन मान लीजिए लेफ्ट से राइट हो रहा है, तो
हाँ.
उसी टाइम ये भी लेफ्ट कोऑर्डिनेट कर रहा है या नहीं कर रहा है?
हाँ, तो कनेक्टिविटी एक्चुअली डायरेक्शनल होती है, बट हमारे जो टूल्स हैं वो ये डायरेक्शनैलिटी यूजुअली पिकअप नहीं कर पाते बिकॉज इट्स सो सटल. बट आप ये मान लीजिए नियरली एवरी रीजन ऑफ द ब्रेन इज कनेक्टिंग विद मोर और लेस एवरी अदर रीजन, बट देयर आर सम वेरी डिस्टिंक्ट वे इट इज कनेक्टिंग. तो इट्स वेरी इंटरेस्टिंग. व्हेन यू स्टार्ट एज अ बेबी, दैट इज एक्चुअली ट्रू. एवरी पार्ट ऑफ द ब्रेन इज कनेक्टेड विद एवरी अदर पार्ट ऑफ द ब्रेन. बट एज यू डेवलप different experiences, it start getting chopped off. Let's say आपके पास एक पूरा जंगल है, but then आप different parts को काटने लगते हैं. What do you call as pruning? Pruning happens then you get a garden. तो different individuals क्यों differ हैं? Because उनकी जो nurturing है that has been different. So very interestingly, babies less than 1 year can identify faces of monkeys and dogs. Can you look at a dog and identify, अच्छा ये face is different than ये face? Can you do that now? No. After 1 year we lose this ability. तो but before 1 year बच्चे ना can differ. अच्छा ये is a different dog than this. This monkey is a different monkey than this. They can identify nuances in languages. जैसे if you know Chinese में there are 4 tones. I cannot do that, but you can, if you listen to it, it will be very hard to make sense of it. ये क्या है? If you're in from certain parts of India, you might use some consonants that other people cannot. जैसे there's a thing in Marathi and even like other South Indian language. It's a thing between la and la. Any Marathi people here? How do you say it? Any non-Marathi people? Non-Telugu? Maybe somewhere from Hindi-speaking belt. What did he say? Not Rida. Mike. Exactly. So very interestingly, these sort of sounds we start to miss after a certain period, 1 to 2 years, because the connectivity has pruned in a way it's no longer perceptible. Japan में they cannot distinguish between ra and la. Ra and la they cannot distinguish. So for them, you say light or right, they'll get confused. You click the right light, they'll be like, they say you click the light. So any Japani person you can say they will be very confused. ऐसे similarly there are like some Indian languages which are these जैसे हाँ, जैसे my name Tripathi, the tha, people in like my colleagues do not speak. Like what is it tha? And even like bha, so they can speak ba, they cannot speak bha. And so, but for us it's very clear. So all this connectivity because of our nurturing has evolved in a way, it has changed the way your brain is structured. and then limited the way in which you can perceive the world. So once we kind of establish this connectivity, we can then make sense of what activity is there. So we define this connectivity by this word called connectome. Connectome is a set of connections between different parts of the nervous system. The connectomes can be micro scale, like we saw for the fly, or can be macro scale, like what we see here. So here what we see is computed using these technologies called diffusion tensor imaging or diffusion weighting. They are water pathways, tracts where water flows. They detect the amount of water signal in these brain tracts and kind of estimate this connectivity. But it's like a highway. It does not tell ki which house is connected with which house. It's like very broad. It's a one share, each share, दूसरे share से connected. But it doesn't tell you which house is connected to which house. So, and then we talk about this thing called functional connectome. It's computed as the correlation of activity of two brain regions. दो brain region, they are going up and down in activity. You say, अच्छा, they are functionally connected. They may not be directly connected. A connected होगा B से, B connected है C से. But A or C are going up and down together, then we can say there is a functional connection. Connectome में जहां पे हम direct signal बिठाते हैं, functional connectome में हम एक indirect signal बिठाते हैं. So the methods that I'll talk about use this functional connectome to make sense of activity in the brain. समझ में आया इधर? Got it? अच्छा 2 मिनट water break ले लेते हैं यहाँ पे to really understand what's happening. नहीं feel तो शायद नहीं हो. अच्छा, now how we compute this functional connectome? तो देखो ये this is of the skull. We collect data from different points. Can you hear me still? अच्छा. And then we do a correlation between each. मतलब what we do a pairwise correlation. इस signal को सारे signal से correlate किया. When we correlate between 2 points, we put a value in this matrix. ठीक है? We correlate between these 2 points, another, तो ऐसे करके we create this value of correlation, and because we repeat it for all points, we have a 2D correlation map. ये समझ में आया क्या? कितनों को नहीं समझ में आया? नहीं समझ में आया? नहीं समझ में आया? ओके, ठीक है. तो आपके पास 10 रीजन है ब्रेन के, उनमें टाइम सीरीज डेटा आ रहा है. एक सिग्नल. So what you do, these two, what's the most common? Like a Pearson correlation. क्या करता है Pearson? दो को एक दूसरे से correlate करता है और एक single value देता है. And what is the range of that value? -1 to 1. -1 का क्या मतलब होता है? उसका क्या मतलब है correlation का? हाँ, जा रहा है तो एक नीचे जा रहा है, उसका मतलब है नेगेटिव कोरिलेशन. अगर 1 होता है तो क्या मतलब? पॉजिटिव कोरिलेशन दोनों एक साथ जा रहे हैं. तो इट गिव्स यू अ सिग्नल बिटवीन -1 to 1, एंड देन इफ यू डू इट पेयरवाइज फॉर one signal to all other signals, you get 9. But then the second with all other signals you get 9, the third with all other signals you get 9. तो ये कंप्यूट करके you meet. You compute yourself, तो ये signal है. Is the value of very small. So you get this matrix, which is again the diagonal is correlation with itself. One, other regions are correlation with other parts. So यहाँ पे आप जो देख रहे हैं, there are some brain regions which are negatively correlated, like the ones in blue, and ones which are positively correlated in red. So there is a discrepancy in how this correlation happens. Some brain networks are more correlated with each other than the other networks. So we can maybe skip. Connectome fingerprinting is a method that we'll talk about. So some prior work has been done on this, which we expanded upon. So we wanted to benchmark this connectome fingerprinting method. It's a computational method, and any such computational method needs benchmarking. ठीक है, अब ये आप लोग कर ही रहे हैं, तो benchmarking के बारे में कुछ सुना ही होगा. कितनों ने नहीं सुना benchmarking? Yes, okay. First of all, to benchmarking, we want to look at space of variables. So this particular technique has a huge space of variables, and we want to pick and choose what space we are working on. So first of all, in connectome fingerprinting, the idea is you make a link between the brain's activity and the brain's connectome. How the activity in one region of the brain and the connectivity of that region that is related. ठीक है, तो उसके बीच में हम लोग एक मॉडल फिट करते हैं. तो दिस इज द क्रक्स ऑफ दैट पर्टिकुलर मॉडल. तो फर्स्ट ऑफ ऑल वी take the activation map of a brain during a task. So here is a working memory task. How many of you know what is working memory? So you're listening to me. There is a small cache in your brain that is collecting this data. जो कि मैं बोल रहा हूँ वो आपके ब्रेन में आ रहा है, एक कैशे फिल कर रहा है, and इसमें you're making sense of things. So that is basically a working memory. We can also do a test here. So let's say, I'll give you a series of numbers. You have to remember these numbers and repeat. ठीक है? Ready? Yes. उठ के बैठना पड़ेगा. हाँ, थोड़ा सा ऐसे ढीले नहीं बैठोगे, ऐसे याद नहीं रहता. 32, 56, 77, 31, 89, 5, 77. Okay, what can you repeat? Who can repeat? Can I have a volunteer? Volunteer? Anyone would like to volunteer? नहीं, कोई भी नहीं. अरे कोई कुछ नहीं हो गया, नंबर नहीं कटेंगे. Okay. Okay. So what you saw is an example of what working memory is. A typical limit of working memory is around 4 to 5 items. Just think. हमको लगता है हमें दुनिया में सब कुछ याद चल रहा है. नहीं. We remember barely 4 to 5 items at a time. And it could be any item. So if you can chunk these numbers, I'm not sure, you guys may be too young. There was a pre-smartphone era जहाँ पे you used to remember numbers. How many of you remember that time? You could remember phone numbers in your head. 10-15 लोगों के नंबर याद रहते थे. अभी तो like अपना भी नंबर I don't know if I remember. And usually the way you remember numbers is a very distinct style. 9717, 562, 584 ऐसे करके. You remember in chunks. So this is called chunking. So working memory, the task that we did would activate the brain in certain way, just like this. So your brain is activated in different parts. So behind, front, and something. Now we want to see if we can model this using connectivity. So connectivity extract करने के लिए we use something called as a parcellation. So it's just a way the brain is divided. मैंने एक मैप लिया और उस पे ग्रिड काट दिया और हर ग्रिड को बोला this is one different region. That's it. अब that grid could be accurate or not accurate. It could be a grid like Chicago or a grid like Paris. ठीक है, maybe correct, maybe not. तो आपने बस एक brain को काट दिया, बोला अच्छा this region is separate to this region, and we use that to create what is called a connectivity map. Second step हमने क्या किया, हमने brain का tissue ले लिया जहाँ पे हम interested है. तो जैसे working memory है, we see a lot of activity in this particular region of the brain. Then we see, okay, let me find, it's very computationally extensive to model the whole brain. We'll just focus on this part of the brain. And then we create a computational model for this part of the brain. How we do it is we create this time series correlation. So you have the resting state data, यहाँ पे कोई भी data you can take. And then you make a correlation between all the, so यहाँ पे, so in a MRI data, you get these things called voxels. Anyone know what a voxel is? It's a 3D pixel. So you have about 200,000 voxels in a brain. A region is a collection of voxels. And what we are doing here is correlating each voxel, each pixel per se, with a region of voxels, the region of pixels. The idea is that that region of voxels would have a similar function. They are going up and down together. They are very correlated, so they are very similar. तो मैं उसको एक region बोल देता हूँ, उसका function similar है. तो we create this voxel cross region को मैं बोल रहा हूँ यहाँ पे parcel. I'm using that term interchangeably. Parcel या region. तो voxel cross region का map correlating each voxel का time series with each parcel का time series. So I get this connectivity matrix. Is this clear? हर voxel का time series एक pixel हो गया of the brain to all regions in the brain which are estimated using this thing I called a parcellation. Parcellation is a set of regions. तो एक set of regions in the brain को extract किया और I create this matrix for a vertices into parcels. Is this clear? हमने यहाँ पे क्या किया, from a very abstract thing as a brain to a very computationally tractable matrix. तो ये बड़ा important है कि from how to a brain to a matrix we got. Is this clear? कितने लोगों को clear नहीं है? Raise of hands, clear नहीं है. क्या clear नहीं है? Yes. So voxels, a parcel is composite of multiple voxels. Let's say हमारे पास इस brain region, इस particular parcellation scheme में 100 parcels हैं. 200,000 voxels, maybe I equally divide. So each is getting how many voxels? There are total 200,000 voxels. मेरे पास 100 parcels हैं, 100 regions हैं. कितने हर किसी को मिलेंगे? Normal divide करें तो 2000, सही? फिर हर इसमें 2000 है, and then find a correlation with each parcel. So 200,000 parcels with each of these 100 parcels. So 200,000 voxels with each of the parcels. ठीक है? तो उससे क्या होता है कि very detailed map बनता है at a level of the voxel region. क्या समझ में आ रहा है क्या? Maybe not. Should I repeat? It's a detailed connectivity map. ब्रेन का एक छोटा रीजन ब्रेन के एक बड़े रीजन से कैसे कनेक्टेड है, सिंपल शब्दों में. ठीक है? इज दिस क्लियर ना? तो वी क्रिएट दिस कनेक्टिविटी मैप एंड देन एसोसिएटेड विद द एक्टिविटी इन दीज वॉक्सेल्स. लेट्स से जैसे आपने 2 बैक वर्किंग मेमोरी करी, तो इस रीजन में आई लुक एट दिस 2 बैक वर्किंग मेमोरी का एक्टिविटी एंड कोरिलेटेड विद दिस पर्टिकुलर मैट्रिक्स, एंड देन आई फिट अ मॉडल and I predict. So this is how I fit a model. I take this data, split into training and testing set. मैं 10 सब्जेक्ट रखता हूँ ट्रेनिंग में, 1 सब्जेक्ट रखता हूँ टेस्टिंग में. और I can do if I have lots of data. मैंने 100 सब्जेक्ट रखे ट्रेनिंग में, 10 सब्जेक्ट रख लिए टेस्टिंग में. You can do 80-20 split, howsoever it's convenient for you. You train a model. This could be any model, and the model is learning the activity in, let's say, the 2 back working memory task and the connectivity in another condition, what we call as a resting state, जहाँ पे आप कुछ नहीं कर रहे हो. तो 2 अलग conditions में, one used to create this connectome, second to make this to associate it with activity. Is this clear? Clear? Yes? No? Confused? Lots of confusion. Any questions around this confusion? So matrix tell us how each region of, how each, so to say, pixel or voxel in this region is connected to all different sets of voxels or different regions in the brain. So it's, yes. Every other region, yeah, including its own, but every other region. So the idea is a very detailed map. How this voxel is connected to the whole brain. Whole brain में हम थोड़ा dimensionality reduction कर लेते हैं because a 200,000 into 200,000 matrix is huge. So उसको हम reduce कर देते हैं 200,000 into 100. और फिर 200,000 को भी रिड्यूस कर देते हैं टू लिमिट इन अ सर्टेन सेट ऑफ रीजन, सो लेट्स से 10,000 into 100. तो उससे क्या होता है कि वी हैव अ मोर ट्रैक्टेबल मैट्रिक्स, अदरवाइज इट्स वेरी कंप्यूटेशनली एक्सपेंसिव. इज दिस क्लियर? यस. कुछ लोग ऐसे मुंडी लाते, कुछ लोग ऐसे मुंडी लाते हैं. इसका क्या मतलब है? कुछ इंडिया में वैसे भी मुंडी लाते. ठीक. So then we make these predictions and see how these predictions are associated with actual data. तो हमने actual data भी collect किया, predict भी किया, और फिर उसे देखते हैं कि कितना अच्छा correlation. Again, इसमें it's very important to the subject out. What we call as, you do not put the same subject in training and testing. Any neuroscience experiment में अगर तुमको वो बोले कि मैंने डेटा लिया और 10 सब्जेक्ट्स में, उन सब्जेक्ट्स में मैंने डेटा X कंडीशन में लिया और Y कंडीशन में लिया, मैंने X में ट्रेन किया और फिर Y पे टेस्ट किया, oh, don't trust it. Subject level data should be separate. Any model that you train should be on a separate set of individuals. You test on a separate set of individuals, because the model can learn individual specific variations, which is not tractable. आप चाहते हो आपका मॉडल है, it should be able to predict out of sample better. नहीं तो आप अगर सब्जेक्ट को रखते हो, it becomes an in sample thing. So we predict out of sample, we use out of sample का testing this thing, and then predict कि अच्छा activity कैसी रही. ठीक है. तो we have a lot of available data sets here. So this is a data set with 1200 subjects and 7 different tasks. Working memory हो गई, emotion task है, motor task है, language task है, gambling task है, and then we see what is happening in different parts of the brain, activity कैसी है, and then उसको we model using, we create this connectome fingerprinting model for all these activities. So हर activity के लिए एक एक model बनाते हैं हम लोग, and then see कि how does that model perform. तो for this example, 3 example subjects लिए हैं, उनका actual brain map ऐसा दिखता है, हमारे predictions ऐसे दिखते हैं. Predictions में यहाँ पे we are showing the amount of correlation. This is again Pearson correlation. So 0.7, 0.84, 0.82, it's pretty significant. Again, unlike machine learning LLMs, you don't get 95% accuracy in brain data. 80% is amazing. So 70 to 80%, good. And then as compared to group, कि तुमने कह दिया कि अरे सबका यहाँ पे activity होता है, our predictions are much better, individual specific. ग्रुप में क्या किया कि मैंने ये बोल दिया कि सबका ब्रेन ऐसे एक्टिवेट हो रहा है, तो उससे मच बेटर है आवर मॉडल, वेर वी एक्चुअली ट्रेन दिस फॉर दिस पर्टिकुलर लाइक सेट, लाइक दिस यूजिंग कनेक्टोम फिंगर. आपने एक मॉडल लिया, उसको ट्रेन किया. ठीक है. देन वी बेंचमार्क इट. तो वी वांटेड टू अंडरस्टैंड हाउ इट इज अफेक्टेड बाय amount of data, motion, different learning methods, and maybe skip some parts of it. So ठीक है. So again, this is some, if you train a model जहाँ पे the people who are doing the task are not doing it well, the model will not train well. जैसे वर्किंग मेमोरी का टास्क आप लोगों ने किया, उसमें अगर आपका एक्यूरेसी 60% से कम आ रहा है तो इट्स नॉट अ गुड टास्क, तो उसका फायदा नहीं है. तो ये नीचे हमने परफॉर्मेंस लिखी. How many of words you remembered out of 7? Let's say that is the performance metric. The better you remembered, the better your model performed. ठीक है. सिंपल शब्दों में. So you want people who can do tasks good. The amount of resting data, state data, जिससे हमने connectome बनाया है, कितना डेटा चाहिए. तो we collect this data in a MRI machine. MRI machine में you go in, you collect data. MRI machine is very expensive. MRI data is very expensive. Do you know how much an MRI session cost? Huh? हाँ, that's an MRI machine, but each MRI session, let's say मैं 1 घंटे MRI में जाता हूँ, how much cost that bear? कितना? 6 to 8,000. Yes. So India में it's around 10,000, but US में it's around $600 or something. So it's very costly. So we want to minimize the amount of data that we need for these models. So what we found out कि our data can start working well with around 25 to 30 minutes of data. Our model. अगर कम होता है तो accuracy is reduced. Another thing, if people go in the scanner and they move, their data is very noisy. It does not train as well. ठीक है. We wanted to look at different algorithms. How do they do if we use ordinary least square, ridge regression, principal component analysis on deep neural network. Again, I'm not sure. AI एक जमाने में ये पहले 3 भी हुआ करते थे. If you don't remember, for a long period of AI, regression used to be a like AI method. Abhi anything less than 20 layers of deep neural network is not considered AI. ऐसा नहीं. एक जमाना था कि you could do pretty well with regression, and what is similar. So we use 4 different methods. One is called ridge regression. Another is called ordinary least squares. Do you guys understand the difference between the two? What is ordinary least squares? OLS क्या है? Anyone? Yes. L1 L2 समझते हैं सब. क्या होता है L1 L2? So L1 L2 regularization में काम आता है. तो L1 L2 norms होते हैं. And then ridge में क्या होता है कि we do a regularization with the L2 norm. तो वो क्या करता है कि आपके जो कोफिशिएंट होते हैं उसको रिड्यूस करता है. OLS में एक चैलेंज है जो रिज रिग्रेशन में नहीं है. हाउ मेनी ऑफ यू नो अबाउट दिस? Maybe not. So all regression this thing suffer from either bias या variance. किसी को एक को प्रेफर करते हैं. तो जब आप रेगुलराइज करते हो आप क्या रिड्यूस करते हो? पीपल आर माउथिंग. बोल के देखो. हाँ, तो बायस. तो we reduce the bias in the model. Overfitting भी bias होता है because it's biased towards the testing data. तो बड़ा इंटरेस्टिंग है कि different even PCA ordinary least square ridge split initial we see some difference in these models, but beyond a number of subjects that difference kind of goes away. तो अगर आपके पास कम subjects हैं तो there are certain methods which is called the leave one out cross validation approach. It works better. Leave one out में क्या? Let's say आपके 10 सब्जेक्ट है, आपने एक हटा दिया, 9 पे ट्रेन किया, 10वें पे टेस्ट किया, फिर यू रिपीट. एक पे टेस्ट किया, 9 पे ट्रेन किया, you get 10 different models. तो वो those 10 different models का sum is something which gives you this, so to say, better accurate model when you have less number of subjects. जब अगर सब्जेक्ट ज्यादा है तो कोई भी ट्रेन कर लो, it's more or less. Very interestingly, people love deep neural networks a lot. For brain data, deep neural networks do not outperform ridge regression. Computationally very expensive, not much use. So यहाँ पे we have accuracy on the deep neural network. It's a 4-layer network around 650,000 parameters something, and there's a ridge on the x-axis which is just 400 parameters. So our model on a model kind of had very similar accuracy. So the brain is somehow still very linear. We don't get a lot of non-linearities that deep neural network can learn from. So if you're wanting to fit brain data, deep neural network की इतनी जरूरत नहीं है. ठीक है. Ridge regression will work most of the time. So this is a summary. Then we wanted to look at test retest. Test retest क्या है कि आपने कोई चीज इस साल करी, आपने उसे एक साल बाद रिपीट करी, should be similar. That is what we want with test retest. Our model had a fairly accurate test retest across all the different tasks. We also wanted to look at, so different MRI machines have a different way that data gets computed. So we wanted to see if we can do like cross scanner predictions, और वो भी काफी अच्छा आ जाता है in this model. So the model is able to learn something inherent, something intrinsic to individuals, and there's not much difference in cross scanner and within scanner predictions. ठीक है. So this was the entire benchmarking fingerprinting thing. ठीक है. अभी यहाँ कोई सवाल है क्या? नहीं. ठीक है. Okay. Then again, this is an extension of the previous work. There's a region in the brain called cerebellum. How many of you know what the cerebellum is involved in? Anyone? Can you guess what the cerebellum is involved in? Okay. Maybe involved in sleeping as well. It's involved in nearly everything. So we wanted to test, we can train the model of the cerebellum. So the cerebellum is a very small region in back of the brain. but it's actually we still don't know what cerebellum does, but it does everything that a brain, the big part of that brain, which is the cerebrum, does. So this is a flattened map. It's like a butterfly. It's cerebellum ko aise khol diya, and this is we wanted to look at how is the. We do a similar thing. We train this model, the same process, and wanted to see how it looks like. We can probably skip this. Our model does pretty well and does a pretty good accuracy and all. Okay. Very interestingly, the model coefficients are very similar to the model activations. Brain का जो activity है और जो coefficient है, they are converging. Somehow the model is trained in a way that is converging on how these coefficients look like, which is an aspect that these particular coefficient, मतलब ये जो brain का, it was not even part of the model. Cerebellum से आपने connectivity ली different parts of the brain की, and somehow this is associated. So the idea is that the coefficients are converging to a base truth of the brain. We still are in discovering कि वो कैसे है, but connectivity और activity में एक बहुत deep link दिखा रहा है because these coefficients are so. Anyway, we skip this. So now we wanted to look at, can we, again, this is AI for social good. We want to see if we can do some social good. So we wanted to use connectome fingerprinting for pre-surgical planning. And hope ये किसी के साथ ना, but do you, any one of you know someone who had cancer? Yes, very prevalent disease. बहुत लोगों को होता है. How many of you knew people with brain cancers? Brain cancer is slightly rare, but it does happen. तो brain के there are some parts जिसमें एकदम cancer से फैलने लगता है. तो उसका solution क्या है? What can we do? Anyone? ब्रेन में कैंसर है, क्या करोगे? काट दोगे. सही, एकदम सही. तो काटते जब हैं तो आपका यू स्प्लिट अपाउट द ब्रेन. तो न्यूरोसर्जन काम करते हैं, ब्रेन का वो निकालते हैं, स्कल निकालते हैं, दे टेक आउट द टिश्यू. बट इसमें चैलेंज क्या है? फर्स्ट, अंडरस्टैंडिंग वेयर द टिश्यू, द ट्यूमर इज. सेकेंड, आसपास के रीजन नहीं काट दे. लेट्स से आपका एक ट्यूमर मोटर कॉर्टेक्स में बैठा है, वो निकाल रहे हैं और आपका हाथ मूव करने का निकाल दिया, अब हाथ ही नहीं मूव हो रहा है आपका. राइट हैंड मूव करने का निकाल लिया, लेफ्ट हैंड से करते रहो, राइट हैंड चल ही नहीं रहा है. तो यू हैव टू बी वेरी केयरफुल. द न्यूरोसर्जन इज वेरी केयरफुल इन टेकिंग कि मुझे बस इतना ही टिश्यू निकालना है, मैं कुछ एक्स्ट्रा ना निकालूं. तो हमने फिंगरप्रिंटिंग में क्या देखा था? एक्टिविटी और कनेक्टिविटी में लिंक. अभी क्या है कि ये जो पेशेंट है, ये एक्टिविटी बहुत कुछ नहीं कर पाते क्योंकि दे ट्यूमर्स ना. तो क्या हम भी कनेक्टिविटी निकाल के यूजिंग समथिंग एज सिंपल एज मूवी वॉचिंग और समथिंग जिसमें बहुत इजी है, कुछ नहीं करना उन्हें, हम उनकी एक्टिविटी प्रेडिक्ट कर दे अराउंड द ट्यूमर, और दैट कैन हेल्प कि इफ लाइक वी डोंट एक्सट्रैक्ट एक्स्ट्रा टिश्यू. So this was done in collaboration with our colleagues at Harvard Medical School. So gliomas, there's a specific form of tumor which is called a glioma, or we use this fMRI to kind of make these predictions. So we had, what we did in this very interesting, we took data from one data set, 150-200 लोगों में, and then used it to predict in these people with tumors, and we did a very very significant prediction. और interestingly इन लोगों के उसमें बहुत ज्यादा डेटा था नहीं, फिर भी हम लोगों ने एक अच्छा prediction किया. तो so this is on the left is the actual activity of when you move a left hand. चलो सब करते हैं, move your left hand. पूरा हाँ बस, very nice. तो अभी आपने ये किया, तो when you move this left hand, this particular brain region in your brain is getting activated. Can you guess which part of the brain it is? Left or right? Oh, very nice. Everyone knows. Nice. So left hand आप move करते हो, आपके right part of the brain में activity होती है. तो in our model, using 60 minutes of data, was able to predict very nicely with a 81% accuracy, 0.81 accuracy. And then just with 4 minutes of data, we were still able to do a significant prediction. तो हमको कितना डेटा चाहिए? तो we kind of realized for predicting after again coming back to the anything after 20 minutes is good, but number of subjects if you have around 20 training में, तो अगर आपने मॉडल 20 सब्जेक्ट पे ट्रेन किया है तो it starts to do pretty well. और कितना डेटा ट्रेनिंग में चाहिए? जब like 4 से 5 मिनट का डेटा, if you are training at least 20 subjects, you start to get very good results. So 20 subject, 5-10 मिनट का डेटा start giving good result. We can split this with this different parcellation scheme. I talked about this. So even different parcellation scheme, the way you cut the brain and define these regions can also have an impact. Some schemes are better than others. Just that. And also how much part of the brain we are looking at, how much extent are we looking at, all that does go into the performance accuracy. अगर हमने ज़्यादा tissue size ले लिया है तो performance may not be, may be better, may not be better. It really depends upon the task here. ठीक है. And also looking at how again मैंने जैसे बोला कि कितना अच्छे से आप टास्क कर पाते हो, जितना अच्छे से आप टास्क कर पाते हो उतनी अच्छी आपकी प्रेडिक्शन एक्यूरेसी हो जाती है. And again, as I mentioned in the previous, we do pretty significant cross-scanning predictions. This is very interesting. So this person had a tumor around the motor cortex here. Can you see? Tumor समझ में आ रहा है क्या? There's a thickening of slightly, यहाँ पे आपका brain का slightly thick हो गया है, and it's sort of a small tumor. तो वहाँ पे we made these predictions of when you may, again, if you move your right hand, left hand, move your tongue, right foot, left foot, तो उसमें कौन-कौन सी activity होगी, we will to very significantly map out how different activities are predicted in this patient. Hello. So, and then with another question, can you see here the tumor? यहाँ पे दिख रहा है क्या? Is it too small? नहीं. Can you see this? ट्यूमर ऐसे बहुत साफ नहीं दिखता है. It's slightly darkened here. That's why you need radiologist, you know. So even with this tumor, we were able to significantly, we were able to make good prediction, but it's not that good. It's slightly lower. So again, the interesting thing is कि if we don't have the ground truth, we can also make these predictions. So इस patient में यहाँ पे एक tumor है. ये देखो यहाँ पे हल्का काला काला है. And even in this patient, there's a small tumor here. In these patients जहाँ पे we don't have the ground truth, we can still make these very strong इंडिविजुअल स्पेसिफिक प्रेडिक्शन. आपका राइट हैंड आप मूव करते हो तो यहाँ पे होगा आपके लिए, किसी और के लिए यहाँ पे होगा. ठीक है, the other side. But so we were able to make these very individual specific prediction, and that we are hoping can help the neurosurgeon make informed decisions. ठीक है. This is a summary of this thing. यहाँ तक समझ में आया क्या? Last part of my talk, but clear till here? Any questions? नहीं? Okay. So in the last part, we use a similar model of modeling the connectome and then associating it with not brain activity but brain dysfunction. So in your brain, if you know people who had Alzheimer's के बारे में पता है? क्या होता है Alzheimer's में? Yes, you have memory loss. ठीक है. So interestingly, Alzheimer's का जो progression है, it starts way ahead when first symptoms occur. You see a person forgetting things in their 60, their symptoms might have started around 45. Brain has this cognitive reserve. आप 45 में कुछ भूलते रहे हो, you start to recover things, but there's a time come एकदम से cognitive abilities आपकी decline करते हैं. And when that time comes, like, then you go to the doctor and see it's all late. So we wanted to see, can we model using this connectome technique of, can we model Alzheimer's? So isme we had this very interesting data set. We had this cohort from this place called Medellin. How many of you heard of Medellin? Colombia, Medellin. Narcos नहीं देखा क्या? नार्कोस किसी ने देखा है? किसी ने? अच्छा, एक बस एक पर्सन दो. That's it. No one has seen Narcos. Very young. So Colombia, Boston was a project, Colbos project कहते हैं. People from Colombia, there's a group in Colombia, there's a family of 5,000 people which have this gene, which is called presenil one, name doesn't matter. But इनमें क्या होता है कि अगर वो gene express करता है, the chance of getting Alzheimer's increases to 100%. तो बस आप ये देखो कि उनमें जीन है, they will definitely develop Alzheimer's. So this population, if we study, we can study how the brain develops with Alzheimer's in like वो Alzheimer's के साथ कैसे develop होता है. It can give us insight on how the brain is developing and how possibly we can cure it. So one of our very good colleagues, she led this project in. She's from Columbia. She's in Boston, so that's why the Columbia Boston project. So to study what we call as autosomal dominant. Autosomal मतलब जिनका genetic disposition है. So it's an early onset Alzheimer's. At the age of 44, they start showing first symptoms, but they have blood markers of Alzheimer's as early as 9, as early as 15. तब से समझ में आने लगता है अरे Alzheimer's का है, ठीक है? So we had 32 such patients and 35 controls, and we wanted to see if we can fit such a model. Again, data set थोड़ा कम रहता है. Again, we can skip this part. Maybe skip this. Okay, very interestingly, on the x-axis we have age, and there's another thing called tau. How many of you know what is tau? No? Amyloid? Speak up. क्या? Mic भाई.
I'm not sure about this tau, but in some sense, like it defines the relaxation time between two certain intervals.
अच्छा. No. So tau and amyloids are brain coagulates. मतलब होता क्या है आपके brain में certain proteins होते हैं, they start collecting and making big chunks with each other. तो वो chunks इतने बड़े होने लगते हैं कि आपका brain में disruption होने लगता है. This is one of the leading hypotheses कि Alzheimer's में क्या हो रहा है कि these tangles of tau and amyloid is creating a havoc in the system. Basically, आपके हाईवे पर बड़े बड़े पत्थर आ गए हैं जमे हुए, और मे भी पत्थर नहीं, उसको कुछ और बोल दो, and it's just blocking everything. So what can you do is remove these. So there are many techniques, but लेकिन वो सब develop कर रहे हैं. So as age increases, the extent of tau, which is here in the x-axis, that increases across different parts of the brain. You can read this. So maybe also this. And brain thickness decrease. So आपका brain क्या है? As these proteins are coagulating, your cell is dying. The brain का जो thickness है वो कम होता जा रहा है. So it's a very strong form of a disease. मतलब हम चाहते हैं कि we have cure for it. So Alzheimer's conference is one of the biggest conferences. So conference on Alzheimer's, about 12,000 researchers go every year. So one of the biggest conference. Okay, we will probably. So this is, we did a connectivity analysis here. We wanted to look at how in healthy, these are the healthy people and these are the control people, the connectome differs. So this is just a from one brain network to another, from one brain region to another brain region का connectome है. अच्छा, why do you, can you guess why it is just one triangle, not the whole? Yes, because it is symmetrical. We are comparing the actual triangles, the data here, and what we see is there are some networks जहाँ की connectivity has decreased and some की increased in people with the Alzheimer's as compared to other people. So using this connectivity approach, you can define कि अच्छा हम यहाँ पे ये difference देख सकते हैं. You can just skip. So now we are going into the modeling part of it, predictive modeling. So in this particular method, what we do is, बहुत छोटा है इसे. Can I zoom? हाँ, ये हो ना बात. Okay. Is this better? दिख रहा है? हाँ. The connectivity matrix we compute for one subject and then we get a behavioral measure. It could be any cognitive measure. So यहाँ पे the measure we use is amount of tau tangles in the brain or amount of amyloid tangles in the brain. The idea is कि क्या मैं इन tau और amyloid को connectivity से correlate कर सकता हूँ. So we then this entire process, maybe I can just skip through. If you, how many of you are interested in understanding this? Oh, social good, very nice. So we have connectivity matrix. We have the measure here, which is in our case tau or amyloid. we correlate each of this measure with each edge. Each edge क्या, एक सेल हो गया इस कनेक्टिविटी मैट्रिक्स में, सेल से हम कोरिलेट कर, अभी भी ठीक है. And then what we see is we find the most significant edges. आपने 20 सब्जेक्ट लिए, हर सब्जेक्ट का आपने मैट्रिक्स एक के बाद एक स्टैक किया, कनेक्टिविटी मैट्रिक्स. आपने हर सब्जेक्ट के लिए एक सिंगल वैल्यू ली, वो सारी सिंगल वैल्यू आपने एक अक्रॉस द मैट्रिक्स हर किसी से कोरिलेट करी, एंड देन यू सी, यू फाइंड द कोरिलेशन वैल्यू एंड देन यू सेव इट हियर. ये समझ आया? Is this part clear? You're correlating, let's say, amount of tau in each individual with their connectivity. तो connectivity between the range, अगर कोई brain region है जिसकी connectivity बढ़ जाती है because वहां पे tau है, तो वो हमें clearly दिख जाएगा. And because this is across subjects, we can see, अच्छा, across subject very stable region है जहाँ पे we can associate the connectivity with कनेक्टिविटी और टाओ या फिर एमोलॉइड का हमने यहाँ कोरिलेशन निकाल दिया, सबसे सिग्निफिकेंट कोरिलेशंस को लिया और उनको वी समड इट अप अक्रॉस अ सब्जेक्ट. तो लेट्स से यहाँ पे हमारे पास 50 पॉइंट्स आ गए, तो हमने 50 निकाल लिया. एक सब्जेक्ट में 60 आए, उसमें 60 निकाले, एक में 70. तो व्हाट इट इज गिविंग, द नंबर ऑफ कोरिलेटेड एजेस, द नंबर ऑफ सिग्निफिकेंट एजेस, द नंबर ऑफ रीजन जो बढ़े हुए हैं इस पर्टिकुलर सब्जेक्ट में. And then we fitted a model, very simple linear model, between this tau या amyloid या behavioral value and this brain measure computed using the sum of these edges. तो basically क्या किया, brain का measure लिया, उसमें significant क्या है, मतलब in the sense कि brain में वो regions which are getting significantly affected, वो हमारा brain का measure हो गया. हमारा दूसरा measure हो गया कि टाउ या एमुलॉइड वो कितना चेंज हुआ है, एंड देन उसके बीच में हमने एक मॉडल बिठाया और फिर हमने लेफ्ट आउट सब्जेक्ट्स में ट्रेन किया. तो वंस वी काइंड ऑफ ट्रेन दिस मॉडल इन लेट्स से वन डेटा सेट, नाउ वी कैन इंक्लूड न्यू डेटा सेट्स एंड स्टार्ट मेकिंग दीज प्रेडिक्शंस. क्लियर है ये पार्ट? समवॉट क्लियर? 50% क्लियर? बिल्कुल क्लियर किसको नहीं है? बिल्कुल क्लियर है क्या? अगेन वी कैन कम बैक टू इट. ज्यादा नहीं है, ठीक है, चलो we'll come back to it. तो our model did decently well. So tau we do significant predictions, and the network, the brain connections which are involved in these predictions are here. तो brain का एक temporal cortex होता है इधर और इसकी connectivity with इधर, that changes a lot in people with Alzheimer's, more tau. Similarly amyloid, maybe we see similar, but amyloid के predictions are slightly less. Similarly, we can also predict behavioral scores. So people जिनकी मेमोरी कम हो गई, अगर उनको मेमोरी टेस्ट देते हैं, हम वो भी प्रिडिक्ट कर सकते हैं कि आपका मेमोरी टेस्ट में कैसी परफॉर्मेंस हो रही है. तो people who are doing pretty well, we can also predict them, and those who are doing pretty poorly, we can predicting using these connectome. We can probably skip this. Parts of the subcortex are also involved. So, again, I'll go to the overall summary. Connectivity based techniques can predict this network organization in healthy subjects. It can make predictions in motor networks in clinical populations. Some work is needed. And also in Alzheimer's, we can make pretty strong predictions using this connectivity model. All good here pe? Okay. So the idea of this talk was to kind of give you a glimpse into it. There might be some aspects which are not clear, but the overall is connectivity ली, उसको associate किया either brain activity से या brain disruption से. Brain activity compute करी using different tasks, brain disruption compute किया using PET, tau या amyloid or something else. और फिर idea is कि if you can train these models, now can you go into, let's say, for a periodic checkup, 10 मिनट 15 मिनट आप हमारे स्कैनर में बिताएं, ये सारे मॉडल जो आपने ट्रेन कर रखे हैं, you those models in this data set and see, can I flag your data? अभी क्या है, these some changes are very subtle that cannot be seen by the doctor, but can we have these models that can make this prediction to say कि अरे अब आप जाके देख लो, आपको ये और टेस्ट कराना चाहिए. So we want to go to a place where we can have a single test or a single way to actually see कि brain is functioning well or not. Make sense? And this is how this entire connectivity predictive modeling space is building upon. Okay, so what we do, my lab is called Integrative Neuroscience and Technology Lab. We integrate neuroscience and technology. We also call it the INT lab. So we do work at this very intersection of it. Now, in case some of you are interested, there is this EEG Foundation Challenge, which is coming up. It's part of the NeurIPS workshop. And NeurIPS, how many of you know NeurIPS? Okay, one of the biggest conferences in machine learning now, AI now. It was a conference for neuroscience. It became a conference for AI. Kya kar sakte hain? But it's a challenge there. And so they have a lot of EEG data and they want to do something similar that we've done with fMRI. Make predictions using one data, test it on another data set. Okay. So if any of you are interested in working or knowing more about this, want to have a fun jab at this, let me know. We can maybe do something. This is my lab at Boston University. Fun people. This was a fun day during COVID. We climbed what we called as Mount Monadnock in New Hampshire. 3.5 hours of climbing. 2.5 hours of coming down. Me and Pallavi, my wife, will never know. Kare, kyun kiya yaar? Humne samajh hi nahi aaya. So Pallavi like, is this your way of doing fun? So Western part of the world, Europe, US, their fun is hiking. If you're going anytime, hiking is not that fun. Okay, questions. Yes.
So, given a time series data, like let's suppose till timestamp t, you generate the similarity between the samples, right? So, my question was like, it gives a scalar value,
Yes.
right? -1 to
-1 to 1.
but it doesn't capture, like let's suppose for the timestamp t, certain part is not that correlated.
Yes.
Maybe the correlation is 0 or something.
Yes. Yes.
But a certain window is highly correlated.
Yes.
Right. So wouldn't it be a better metric to have a vectorized or a windowed correlation that captures a better correlation among
Yes,
the samples?
there are few people who have done that. They called it very funnily connectome. They did like each time sample pe what's happening and they did a correlation.
But
But
each time sample will be very fine, right?
yes,
So
we can do a moving window or something.
Right.
They did something kind of that. Usme na, there are some points which can be very noisy.
Okay.
So MRI ke saath challenge hai, as I mentioned, so MRI क्या है? It's a blood oxygenation signal. तो ब्लड में जितना ऑक्सीजनेशन जा रहा है तो आइडिया है कि जहां पे ब्रेन एक्टिविटी है वहां ऑक्सीजनेशन ज्यादा होगा. बट इफ यू मूव, if you take a deep breath, if your heart rate has increased, that changes it a lot. So there are very noisy patches of it, and as you rightly said कि Pearson correlation का limit क्या है कि outliers drive the relation.
Right.
Let's say you have 2 time points which are very significantly high, maybe because of one signal, some noise. That signal becomes very correlated. So we do this noise cleaning. We correct a lot of this noise, but again there could be some signals. But it's an interesting idea. Some people have tried it. It is computationally expensive, very extensively, and but could be done. Anything?
So, sir, how can I get more information about that NeurIPS workshop challenge?
About what?
That NeurIPS workshop challenge.
Oh,
that you
NeurIPS
talked about.
workshop challenge. So just go to EEG Foundation challenge. If you want to do it by yourself, free. If you want to drop me a mail, you can check my website. Reach out to me if you want to do more. Again, it'll be a lot of work. It's a lot of data, lots of analysis. बहुत मेहनत लगेगी. But yeah, if someone is interested, just ping me.
Right. So I'm actually working on glioma classification on the basis of the WHO grades. So it's classified into grade 1, 2, 3, 4. Right. So I was wondering if this kind of thing can be done there as well, the connectome fingerprinting thing.
You need to have data for those individuals.
Yeah, I actually do have data. I'm working with Ames Library.
Then do you have only the structural data? Also, do you have the fMRI data?
I do have MRI data. So I have MRI data for some 30-ish patients with different sequences.
So MRI there are two types of MRI data. One is called structural, one is called functional. So if you have functional MRI data, then this could be done.
Okay, how do I differentiate between them then?
Structural, you can just ask, is it structural MRI or functional MRI? So you can just like say, fMRI data hai ki nahi. fMRI data is when doing something, they are doing something functional. Let's say they're doing like hand movement tasks, working memory tasks, or doing nothing in the scanner. So it's a different MRI paradigm. Structural paradigm में आपको एक snapshot मिलता है brain का with how the brain is structured. कैसे यहाँ पे ये tissue है, उसकी ये thickness है, that overall image. But MRI is kind of a time series signal, fMRI. So that is it. And then
okay, thank you, sir.
Available, that could be possible. Error should be? Yes. Yes. Yeah, that's why these things are still not implemented in clinic as such. These are still areas of research. We need to be that confident we can say ki, well, that's why we want to increase our accuracy as much as possible. Yes. You don't want to damage and like on ethical grounds, is that correct or not? But the thing is that what's the other solution? The surgeon even doesn't know. So abhi kya hai na, the surgeon is making that decision. Surgeon is using this information along with all other information and making that decision. So they are not only relying on our prediction, they are using a set of data and it's like more clues whether this tissue could be cut or not. So we are helping for that. But we are hoping कि ऐसा हो जाए कि we can make very strong prediction, but it's more research is needed. Yes.
Hello, sir. I was just wondering that in parcellation we do rather than clustering regions based on correlated activity. Like in parcellation we cluster regions.
Can you repeat the question?
Sir, in the parcellation technique, we group the regions based on correlated activity, right?
Yes.
So I was wondering if we could group together nodes or the clusters. We make clusters based on causal roles, like if such that like it could affect like regions that have common causes to multiple activities. Like we can.
So causation is hard to determine, especially in neuroscience. There has been a very active debate that how can you find causal relationships between different brains, because this is the thing with the entire field of complex science. Complex science. Network science. Network are so densely connected. What causes what is very hard to say. आपका A, B, C, D, E, F है और उसमें सब नोड एक दूसरे से कनेक्टेड हैं. आप A की वजह से हो रहा है या वो F की वजह से हो रहा है या कॉम्बिनेशन ऑफ A, B, C की वजह से हो रहा है. So it becomes very difficult to find causation. What we can really say is correlation. अच्छा ये correlation हो रहा है क्या? So that's why we are using correlation. But if we know causation, there will be anything.
So you mean to say that if there are there are a lot of confounding variables in this?
Confounds another thing. Determining causation in a complex system, in a system which is so densely connected, is always very hard. Stock market में stock ऊपर जा रहा है नीचे जा रहा है because there are so many players at play. मतलब यूएस की फेडरल उसने थोड़ा सा रेट चेंज कर दिया, इंडिया में कुछ और हो गया. so much variables. Saying कि A determines B is very hard. There are so many factors in play. So that's the same in the brain. No, so many neurons, so many activity happening. So it becomes very hard to say causation truly. That's why we only say correlation.
Okay, sir. Thank you, sir.
Yeah.
So when we were looking the connectivity for say 2 backtasks, when we go for 3 backtasks or 4 backtasks, does this connectivity changes?
So connectivity to stays similar, activity changes. So 2 back tasks, 3 back tasks become very hard as compared to 4 back tasks. Sorry, as you go harder in back, it becomes 2 back is easier than 1 back, 3 back is easier than 2 back, 4 back is I've never done 4 back tasks. पता नहीं कौन करता है. No, it's so difficult. So different tasks will give you different activation map, the way the brain is activated, and then the the association with the connectivity map may differ. Connectivity map, we don't use task to create that. We use something called as resting state, जहाँ पे कोई कुछ नहीं कर रहा है. That gives you this correlation map of different brain activity. Different brain dysfunction. So it's a connectivity which is not using activity data. एक्टिविटी डेटा को लेके हम उससे एसोसिएट करते हैं कनेक्टिविटी का. ठीक है. हम
जेंडर पे क्या डिफरेंसेस होती है? एक मेल का कनेक्टिविटी उस टास्क में और एक फीमेल का कनेक्टिविटी उस टास्क में काफी अलग होता है या थोड़ा सा सिमिलर होता है?
देखो, टास्क स्पेसिफिक कनेक्टिविटी चेंजेस से ज्यादा एक्चुअली इंडिविजुअल स्पेसिफिक कनेक्टिविटी चेंजेस बहुत होते हैं. सो देयर इज activity dependent change, there is like individual dependent change. So is it related to male and female? Could be there. Could be there's a what difference between individuals. So like एक मेल वर्सेस दूसरे मेल, उसमें भी बहुत डिफरेंस हो सकता है. तो उसका एक तो रीजन है द ब्रेन साइजेस आर स्लाइटली डिफरेंट मेल्स एंड फीमेल्स में. Connectivity, activity could be different, but there is so many individual level variations that we don't know yet. Challenge with MRI is the biggest MRI data is just 50,000 people. Saying 50,000 people में how 8 billion people work, it's hard. So we need more and more data. The challenge in this field is again cost, number of people we scanned yet. As data, the more better improved models we can have.
एक और डाउट था कि ये इस फील्ड में नॉर्मली ये दो वर्ड इंटरचेंजबली यूज होते हैं. जैसे आपने कहा कॉग्निटिव स्टेट्स, फिर कोई वर्ड इस्तेमाल होता है मेंटल स्टेट्स. क्या दोनों काफी अलग है या
No,
similar?
I'll think use interchangeably. Yeah. You can use cognitive state or mental state.
Okay, thank you.
Done? हो गया. 1.5 घंटा हो गया. Okay, then I hope you learned something. I hope neuroscience is something intriguing. I was not expecting you to get everything, but थोड़ा सा maybe neuroscience और explore कर सकते हैं. So there's a lot of computation in neuroscience. So I urge people who are willing to do a lot of computation. There's a lot of scope. And yeah. Any other questions you can mail to me, or if you're here for a few days, maybe you can bump into each other. Okay. Thank you.